Have you ever picked up a prescription and just wondered, how did this little pill get here? What kind of epic journey did it take to go from some scientist's wild idea all the way to your local pharmacy? Well, it's a long, complicated, and incredibly high-stakes process. And today, we're going to break it all down.
This right here is the single most important question. I mean, think about it. We desperately need new cures, new treatments, new hope. But at the same time, the first rule of medicine is do no harm. So how do you balance that? Well, the entire system we're about to talk about, it was all built to answer this one critical question.
So why do we have this massive, complex system of trials in the first place? With all the rules and regulations. It wasn't always like this. The truth is, this whole process was born from some really dark moments in medical history. Those mistakes forced us to build a powerful ethical foundation for any and all research involving human beings. Yeah, pretty much every major step forward here was a direct reaction to a tragedy. The Nuremberg Code, that came right after World War II, laying down the absolute basic human rights for people in research. Then, the Declaration of Helsinki took those ideas and made them global for doctors. And here in the US, the absolutely shameful Tuskegee experiment led directly to the Belmont Report, which gave us the core principles: respect, doing good, and justice that guide everything today. And all of that history brings us to maybe the most important safeguard we have: informed consent. Now, this is so much more than just signing a piece of paper. It is a mandatory, in-depth process. It's where a potential volunteer learns everything: the good, the bad, the risks, the potential benefits, what other options they have before they ever agree to participate. And they have to be crystal clear about a few things. The form has to state, flat out, this is research. It is not a guaranteed cure. It has to make it obvious that you can walk away at any time, for any reason. It is 100% voluntary. It has to explain how your privacy will be protected, and it must give you a direct contact for any questions you have. It's all about putting the power in the hands of the participant. Okay, so with that ethical groundwork laid, let's actually walk through the journey. The whole thing is broken down into 4 main phases, and each one is designed to answer a very simple, very specific question. Think of this as our roadmap for the next few minutes. Now, before a new drug gets anywhere near a person, it has to spend a long, long time in the lab. This is where the story really begins. These are the preclinical studies, and yes, they often involve animals. The whole point here is to get the first clues about safety, to figure out the basics of how the drug even works, and to understand what our bodies do with it, how it's absorbed and flushed out. You have to have all this data before you can even ask for permission to start human studies. And if it passes that stage, we move to phase 1. This is the very first time the drug is given to people, and it's usually a really small group of healthy volunteers. And listen to this, the main goal isn't to see if the drug works. Nope. The only goal is to find the highest possible dose that people can take without getting serious side effects. It's all about safety, period. All right, so once we know a drug is basically safe and we have a dose range, the big question shifts. Now in phase 2, we finally start looking for the first real signs that this thing might actually work against the disease. And to get those signs quickly, researchers often use a clever shortcut called a surrogate endpoint. So, for instance, instead of waiting 10 years to see if a drug prevents heart attacks, you might just measure if it lowers blood pressure. It makes sense, right? It's a faster, cheaper way to see if you're on the right track. But here's the plot twist. That logical shortcut, that thing that seems to make perfect sense, sometimes it can be completely, utterly, and tragically wrong. Relying on these unproven shortcuts can lead to absolute disaster. Let me give you a famous example, the Cardiac Arrhythmia Suppression Trial, or CAST. The thinking was so simple, so logical. We know that abnormal heartbeats after a heart attack are linked to a higher risk of death. So, a drug that gets rid of those abnormal heartbeats should save lives, right? Makes perfect sense. It was horribly wrong. The results were just shocking. The drugs worked perfectly. They got rid of the abnormal heartbeats, but the patients taking those drugs died at a rate 2.5 times higher than the patients taking a sugar pill. It's just this incredibly powerful and tragic lesson about the dangers of assuming a shortcut works. And that is exactly why we have phase 3 trials. After a drug proves it's safe and shows some early promise, it has to face the ultimate test, a huge, super rigorous trial designed to give the definitive answer. This, my friends, is the main event. This is where we have to talk about a really key difference: efficacy versus efficacy. See, phase 3 trials are all about efficacy. Does the drug work under the perfect, pristine, controlled conditions of a study? But that's not the same thing as effectiveness, which is, does it work out in the messy, chaotic real world with all kinds of different people? A win here is a huge deal, but it's still not the end of the story. So, how do you make sure the test is actually fair? The absolute gold standard is randomization. You basically flip a coin for every participant to decide if they get the new drug or the control, which might be a placebo or the current best treatment. By doing this, you make sure the only major difference between the two groups is the medicine itself. It's the best tool we have to get rid of bias. And to fight bias even more, we use something called blinding. In a single-blind study, you, the patient, don't know if you're getting the real drug or the placebo. In a double-blind study, which is the most common, neither you nor your doctor knows. And in a triple-blind study, even the people analyzing the data are kept in the dark. It's all about making sure the results are as objective as humanly possible. So if a drug makes it through all of that and passes phase 3, it can finally get approved and go on the market. You think that's it, right? Story's over. But nope, the learning never, ever stops. Phase 4 is like a never-ending watch party that happens once the drug is out in the real world. And here's why that is so incredibly important. Even after all those years of testing, about 4% of drugs that get approved are eventually pulled from the market because of safety problems that only show up later. The simple fact is, even a big phase 3 trial isn't big enough to catch those rare but sometimes very serious side effects. This ongoing surveillance depends on doctors and patients reporting any problems they suspect are linked to a drug. And as you can see, that reporting has gone way, way up. This constant stream of real-world data is absolutely essential for us to build a complete safety picture of a drug over its entire lifetime. Now, throughout this whole journey, from the very first person in phase 1 all the way through phase 4, there are independent referees watching over everything. You have institutional review boards, or IRBs, at the local level, and then you have data and safety monitoring boards, or DSMBs, that are constantly peeking at the results of ongoing trials to make sure that patients are being kept safe. So there it is. It's a slow, expensive, but incredibly rigorous and ethically driven system. It's really our best tool for balancing that desperate need for new medicines with the absolute, non-negotiable need to keep people safe. But it's not perfect. And in a world that seems to need cures faster and faster every day, it leaves us with one heck of a question. How can we make this process better and faster without ever sacrificing the safety that all those historical lessons